Okay, Dr. Asejo.
Hello, everyone, and welcome to today's Grand Round session. Today is my pleasure to introduce Dr. Aman Chauhan, and I'll tell you a little bit about him. He earned his medical degree from the Kasturba Medical College in Manipal, Karnataka, India, followed by a dual residency in internal medicine and pediatrics at Louisiana State University, New Orleans. Dr. Chauhan completed his fellowship in hematology and oncology at the University of Kentucky, especially focusing on neuroendocrine tumors. Additionally, Dr. Chauhan completed a cancer therapy evaluation program, the CTEP, physician externship at the National Cancer Institute in Bethesda that focused on designing clinical trials and clinical research projects. His clinical interests include treating neuroendocrine tumors, including carcinoid tumors, high-grade neuroendocrine carcinomas, and small and large neuroendocrine carcinomas as well. Dr. Chauhan leads the University of Miami Neuroendocrine Cancer Program and co-leads Sylvester Theranostics drug development program. He's national principal investigator on several investigator-initiated neuroendocrine cancer clinical trials. He has authored over 70 scientific publications and book chapters and has received Career Development Award from the NCI CTAP program. Dr. Chauhan also serves on the American Joint Committee on Cancer and the American Society of Medical Oncology NET Guideline Committees, and is an active member of the NANET's Communication Committee as well. Dr. Chauhan is board certified on internal medicine and medical oncology. He's a member of the American Society of Clinical Oncology, as well as the American Association for Cancer Research and the North American Neuroendocrine Tumor Society. Again, it is my honor to introduce Dr. Chauhan, who will be speaking on the interesting topic of theranostics and drug development. Thank you, Dr. Chauhan, for joining us today.
Thank you, Dr. Asejo, for a warm welcome. Thank you, Dr. Weiss, for having me here. It's always a pleasure to interact with my colleagues in dental medicine. My name is Aman Chauhan, I'm a medical oncologist here. And today, I will be discussing this relatively newer field of medicine called theranostics. We probably didn't hear about it a lot, maybe 4 or 5 years back. And from having minimal representation in drug development, now this field is expected to grow from current $2 billion footprint to about $7 to $8 billion in next couple of years to about $25 billion in this coming decade. So this is a big area for both drug development, diagnostics, and has a huge untapped potential. And today I will kind of go a little bit into what theranostics is and also acquaint some of our colleagues with drug development that we are personally doing in this particular space. These are some of my disclosures. So I'll start off with my area of clinical interest. So besides radiopharmaceuticals, my main clinical and research interests focus on neuroendocrine cancers. And neuroendocrine cancer is one of those rare diseases, not one disease, a spectrum of disease. Although relatively rare, we have seen a huge surge in the incidence of neuroendocrine cancers. While most cancer, our incidence is relatively stable, and in fact declining in last few years for common cancers like breast cancer, lung cancer, colon cancers. In the same time frame in last 50 years, we have seen about 8-fold increase in the incidence of neuroendocrine cancers. And what's even more worrisome is the cumulative prevalence of neuroendocrine cancers. They are technically rare based on incidence. However, our patients go on to live, years to sometimes few decades. So the cumulative prevalence for neuroendocrine cancers have really catapulted into the second most prevalent neoplasm of GI origin after colorectal cancer, so much higher than pancreatic, adenocarcinoma, gastric cancers, and hepatobiliary cancers. All of this growing burden of neuroendocrine cancer disease. And then, I'd like to mention there are over 170,000 patients diagnosed with diagnosis of neuroendocrine cancer in the US alone. So this growing burden of neuroendocrine disease in the US and globally has really focused attention into it in last decade, and we have seen because of that there has been some acceleration in drug development in this area. uh, it wasn't too long back when I was a resident in LSU, that we had to send patients to Europe and sometimes to Asia for some of the treatments, because once they progressed on somatostatin analogs, there was really nothing on label for many of these patients. However, as you've seen, last 5 to 7 years, there's been slew of FDA approvals, and we are getting better, and the patients outcomes are improving. The latest kid on the block, the most recent FDA approval for therapeutics, the lutetium-177 dotatate, it's a radiopharmaceutical, it's a theranostics drug. And that's where I will pivot to our topic today of theranostics. This is a cover page of the Lancet Oncology, very prominent journal in the space of oncology. And they put theranostics on the cover page just to kind of highlight growing importance of theranostics. And so what is theranostics? It's really a technology where we use and combine both diagnostics and therapeutics. And that's where the word comes into theranostics. Some people like to use theragnostics. We add G for diagnostic and therapy. So what do we mean by that? If we have a target on a cancer cell, right? For example, let's take neuroendocrine tumors. And neuroendocrine tumors tend to have somatostatin receptors on its surface. So if we have a target and we have an analog, right? Somatostatin analog. So if we inject that analog, it will go and find the receptors on the tumor. So if there is a way to link a radio tracer to this analog, we can essentially track where the tumor is in the body. And that's what we do with some of our PET scans. We have gallium-68 dotatate scan, basically link gallium-68 mild radio tracer to somatostatin analog, inject that radio tracer, and then take a image, and we can see exactly where the tumor is. And if patient has somatostatin receptor positive disease, all we need to do is now flip this radio tracer to a more heavy duty anti-cancer radio tracer, like lutetium-177. So it's a beta emitting radioisotope. Same concept, we inject the drug, the somatostatin analog finds the tumor by joining it to somatostatin receptor on the tumor, and then the lutetium-177 attached to that somatostatin analog radiates the tumor and kills the tumor. And this is a precision way or a tailored way to give radiation to the tumors and avoiding normal tissue from radiation exposure. We've now successfully done this in prostate cancer. So currently we have two theranostics that are FDA approved, Lutathera for neuroendocrine tumors and lutetium PSMA, the Pluvicto drug for prostate cancer. However, this is just a start. As long as we have a good target, we can really administer this radio tracers by linking into an analog for that target. And I'm here in the example you see this PET scans from breast cancer, pancreatic cancer, and colorectal cancer is expressing a target called FAP, fibroblast activating protein. So if we can detect them, all we need to do is now flip the gallium-68 to maybe lutetium-177 or actinium to now irradiate these tumors and selectively kill the tumors. And that's where the promise of radiopharmaceutical or theranostics comes in. For my rest of the talk, I will focus more on lutetium-177 dotatate because of its relevance in neuroendocrine space and my personal research in drug development with this. So just to kind of reiterate what I mentioned, what is Lutathera or lutetium-177 dotatate? As you can see here, neuroendocrine tumors on its surface express somatostatin receptors, okay? And the drug Lutathera is lutetium-177, which is the radioisotope. It emits beta irradiation, and it's linked to dotatate, which is a somatostatin analog. Once we inject this drug, it goes and binds to the receptor on the surface of the tumor, gets internalized, and once inside the cell, the lutetium-177 would emit beta ionizing radiation, causing DNA break. Once the DNA has nicks in it is damaged, the tumor cell dies. We know this drug works because it was tested in a large international multi-center registrational phase 3 study. Patient who got Lutathera, so on the green line, the median PFS was not even reached at the time of reporting in this New England Journal of Medicine publication. We now know this is in the range of about 28 to 30 months, as compared to control arm of higher dose SSA was just 8.5 months. This is a fantastic looking Kaplan-Meier curve. We often don't have luxury to see these kind of results for a lot of diseases in cancer space and for a lot of drugs. But this was a resounding success, and based on this, FDA granted his approval. This is one of my patients from my previous institution where we treated this patient on standard of care Lutathera after FDA approval. And you can see here on the left-hand side, that's a gallium-68 dotatate picture of the patient, and hardly any bone in the disease in the patient is untouched by the disease. You can see small and big lesions, axial skeleton and the pendicular skeleton, and liver. So every dark spot is a somatostatin receptor positive tumor. Then we gave 4 rounds of Lutathera, and there's about 66% reduction in the molecular tumor volume. More importantly, clinically, this patient came off narcotics and had really good disease control for a while and good quality of life. So this is now a reality, and we have access to these fantastic drugs, which previously we had to send these patients to Europe and India and other countries and didn't have access here. However, there is more that needs to be done. As I mentioned, the median PFS is about 30 months. Can we improve that further? Can we make it 50 to 60 months? One of the Achilles heel for Lutathera is, at least in the neuroendocrine tumor space, it might be good in stabilizing the cancer. It's really not good in shrinking the disease. Only 14 to 18% of patients shows responses. That means some degree of shrinkage of the disease. So can we enhance the activity of Lutathera and make it with higher response rates? And of course, there are some other unanswered questions. Where does Lutathera fit in the current paradigm? It works really well. Should we all use it up front, or second line, or third line, or fourth line? Now, how can we reduce some of the long-standing bone marrow toxicities with these radiopharmaceutical? Currently, the risk of MDS leukemia is reported under 1%, or 1% range for Lutathera. All right, so this is where we come in, and our research interest focuses on and building on success of Lutathera and trying to make it better, smarter. And the way we are trying to do it is combining it with drugs that complements Lutathera. And we will be discussing some of these trials. Some of my colleagues at other centers are looking at other ways in enhancing PRRT field by testing alpha PRRT or different analogs like lutetium dotatalk, and sometimes even antagonists. So different investigators are kind of looking at different ways to take the field forward. These are some of my investigator-initiated trials in different phases. I'm happy to now tell you that phase 1 trials are now both phase 1 trial of triapine is completed accrual. Phase 1 trial of peposertib, we just put in the last patient. And then the phase 2 have just begun. So we'll take a little time about discussing some of these trials, because these are our own science. We wrote the protocol, we kind of took them through different stages, and kind of are now taking them from phase 1 stage to phase 2. So let's talk about triapine trial first. What is triapine? Triapine is not a new drug. It was first, I think, discovered in Yale, maybe about 20 years back. It's a ribonucleotide reductase inhibitor. This enzyme is critical in building of DNA, building blocks. So converts ribonucleotides into deoxyribonucleotides, right? And if we can block this enzyme with help of triapine, we essentially impair cancer cells' ability to repair the DNA after Lutathera causes DNA damage. So it complements very nicely as a radiation sensitizer. How did we know this? Back in 2018, when I was at NCI, Lutathera was just FDA approved for NETs, and I was looking for a good drug that can complement. And roughly at that time, triapine phase 2 data came out from gynec cancers, where it was used as a radiation sensitizers. And Dr. Kunos at that time had just reported that there was about fantastic durable complete responses, and moreover even a follow-up data in 3 years showed about 80% metabolic complete responses. So it sounded very promising as a radiation sensitizer. We wrote a small grant to NCI to test radiation, the triapine, in our preclinical models. This is one of our radiation oncology resident in Kentucky who did help us with some of the in vitro work. And our in vitro work did suggest that there was synergy, and triapine works as an excellent radiation sensitizers in QGP and Bond cell lines, which are the pancreatic neuroendocrine tumor cell lines. We also then validated these data in animal studies. But fast forward, we were impressed with the activity, and we proposed a first-in-human combination phase 1 trial for NCI, to test triapine along with standard of care Lutathera. So as you can see here, IV bags of Lutathera, which is standard of care, is given once every 2 months for a total of 4 doses. And we administer triapine as an oral pill for 14 days during each Lutathera treatment. Patient could take it at home. And as the radiation kills the tumor over the 2-week period in the patient, triapine sort of adds, as a DNA, it prevents the DNA repair and blocks that process. And hopefully we will see that it enhances the efficacy of Lutathera. That was the premise. Since this was first in human study, the main goal was here to find the right dose for triapine in combination Lutathera. But we are also looking at efficacy signals and some interesting science. Besides that, we biopsied most of these patients. We collected a lot of blood, and then we are currently running analysis, RNA sequencing, NGS, and some novel promising blood-based biomarkers, and studying them, understanding the biology behind responders and non-responders. This was the first patient that I treated, and call it a beginner's luck. Our first patient itself had a partial response, and the patient continues to do really well, almost now, I think, 3 years out. So some success there. Now 30 is over. The study was open, I think, close to 8 centers, Mayo Clinic, Huntsman, City of Hope. So it was truly a national multi-center study funded by NCI. And we concluded, we've now treated 31 patients, and I presented data at AACR earlier this year. And we are seeing significant activity. We've almost doubled to triple the response rate as compared to standard of care Lutathera. And that was our goal. And we've also found the safe recommend phase 2 dose. So this study, it was really gratifying to see it from inception, from our lab to phase 1, now moving on to randomized phase 2. We have just launched the randomized phase 2 study. It will enroll 100 patients. Nationally, we expect it to open in 20 plus cancer centers throughout the US. And we're going to now randomize patients between triapine plus Lutathera versus Lutathera. And that would really give us a good idea about long-term safety as well as efficacy, and before we jump into a randomized phase 3 study. So that was our first study, and turned out to be a success. The other study that we ventured into was a rather novel drug at that time. In fact, it didn't even have a name when I first started working with it. It's called M3814. Now it's called piposertib. It's a DNA PK inhibitor. It's one of the enzymes that is really responsible for DNA repair, especially in the tumor tissues, once the DNA damage has been induced either by radiation or chemotherapy. And this particular target is very specific. So this particular drug does not have any off-target effects. Okay? So that's the premise of this particular drug. It's a fantastic DNA repair inhibitor. Unlike trypene, which by itself also have some cytotoxic activities, a mild chemotherapy, DNAPK inhibitor does not have any activity on its own. You really need to induce DNA damage for it to work. I had just finished my fellowship at that time. This was my first grant that I received. Got the drug. This was brand new drug at that time. It was just being tested in maybe one or two cancers, period, total. Very excited to find, get to work with a brand new drug right off development. We tested it in various models. This is one of our mouse models, for human pancreatic neuroendocrine tumors. I was really blessed to have wonderful basic science colleagues at University of Kentucky, and now we're kind of trying to build those relationships with basic scientists here at University of Miami. I collaborated with Dr. Tadahide, who helped us with the radiation of these models. We completed these experiments within the first year from the grant. And as you can see here, these are clonogenic assays, in vitro modeling. And what we can see here with higher dose of radiation, of course, you expect the cancer cells to die, but there was a synergistic activity with higher dose of trypene, you had induced more cell kill. In vivo experiments, as you can see, when peposertib was added to radiation cohort, there was just fantastic stunting of the tumor growth, while peposertib by itself doesn't do anything, as you can see, because you need to induce DNA damage, right? So this was fantastic results, and led us to kind of bag a grant, a larger grant, to do a multi-center first in human phase 1 study with peposertib and Lutathera. This study was open at 6 cancer centers throughout the US. And again, I'm very happy to announce the study is now concluded. We presented data last month at Montreal for the recommend phase 2. And then after the dose escalation cohort, we had enrolled 14 more patients to understand long-term safety as well as efficacy signals. And I'm happy to announce that as of last week, we enrolled the last patient on this study. So the data for that will have to mature a bit. We'll probably report on expansion cohort next year. And we are kind of thinking of potential ideas where to take this combination forward, perhaps in higher grade neuroendocrine cancers, which are a critical unmet need right now. The other study that I'm co-chairing with my colleague, Dr. Simran Singh from Canada, and is an important study in our field because, as you all know, that Lutathera by itself works really well, but eventually cancer patients progress through it, maybe a year, 2 years, 3 years later. And one of the common questions my patients ask me, doctor, this treatment works so well, can I try it again, right? It's a 4 doses treatment. So it worked so well and kept my cancer at bay for a few years. Can I try it again? It's very, very intuitive, right? However, currently FDA approval for Lutathera is just for 4 doses, one time. So there is no prospective randomized data in support of the retreatment with Lutathera. So we were a little cheeky and we kind of named our trial as Net Retreat Study. So we're going to test the hypothesis that we can safely retreat patients after they progress on Lutathera with limited dose Lutathera, and have a control arm of current standard of care, which is everolimus. If this study turns out to be positive, I'm hoping it will be practice changing and allow us to continue to retreat with some good level of evidence backing that up. It's also a very unique study in the sense it is an international study. It's a joint efforts between CCTG, the Canadian Cancer Treatment Group, and SWOG, the US-based cooperative group. All right, so we talked about targeting somatostatin receptor in neuroendocrine tumors in different ways and trying to enhance our current standard of care therapies. But is there a role of targeting same drug, same target in other cancers beyond neuroendocrine tumors? I think so. And this was one of the projects I did with one of my pharmacy students. At my previous institution, I pulled tissues from biobank and stained them from somatostatin receptor. And what we found is there are a lot of tumors besides neuroendocrine tumors that express somatostatin. And some of the big ones that really caught our attention: small cell lung cancer, which is 14 to 18% of all lung cancer, small cell lung cancer. Again, a very critical unmet need. So there might be role for targeting other tumors like Merkel cell cancers, paragangliomas, pheochromocytomas, beyond just the typical NETs, targeting somatostatin receptors. This is one of my patients who's referred to me from Pittsburgh, esthesioneuroblastoma. Again, not something that I treat in my clinic, but had somatostatin receptor positive tumor, so was referred to me for PRRT, and I did treat him with PRRT and a significant benefit, and this is the patient who had progressed on all potential treatment options, and it was this versus hospice, and then patient had a fantastic benefit. So again, these kind of drugs are really, as long as you have a target, can be safely administered and should potentially have activity because radiation kills anything viable, right? Switching gear for my last segment for the talk, talking about the other type of neuroendocrine cancer that I also see in my clinic. So again, neuroendocrine cancer is not one disease, it's a spectrum of disease, from low-grade typical carcinoid to high-grade neuroendocrine cancers like small cell lung cancer or large cell neuroendocrine cancer. And I'm not even mentioning other rarer subset of neuroendocrine cancers like paraganglioma, pheochromocytomas, Merkel cell cancer, right? So just within the spectrum of neuroendocrine cancer, it's from slow growing indolent disease to very rapid growing fatal disease. Small cell lung cancer, 5-year survival is under 5%. Most patients survive probably a year to year and a half from the time of diagnosis. So again, a very critical unmet need. And as I mentioned, that a subset of high-grade neuroendocrine cancer patients, like small cell lung cancer patient, it do express somatostatin receptors and other novel targets that we can potentially target via Theranostics. So for example, this is a case of somatostatin receptor positive small cell lung cancer, and patient got Lutathera and had a fantastic response. Another study done at Georgetown showed that Lutathera can potentially be applied in high-grade neuroendocrine cancer patients like small cell lung cancer. However, these were early phase 1 studies, smaller studies. We are going to be opening not one, but a couple of studies here in Sylvester targeting somatostatin receptor in small cell lung cancer, both frontline and refractory settings. So we're very, very honored to kind of open these studies, which are probably very select 4 or 5 sites in the country who will have access to those technologies. Besides that, we also are looking at targeting small cell lung cancers and other solid tumors for FAPI, fibroblast activating protein. So we are in the process of activating this study. And then we are also opening other novel radiopharmaceutical studies targeting, for example, integrin, right? And this is a biomarker not only seen in small cell, but various other solid tumors. So just wanted to give you a taste of how the radiopharmaceutical field is evolving as we are getting more comfortable with handling radiation-based drugs, scaling up the product, getting the supply chains intact. Now what we have to do is find the relevant target and formulate the drug to target that particular target, and design smarter studies. And you will see that this field will continue to evolve and get bigger. Before I leave, I want to give a little shout out about our newer program. So I joined University of Miami Sylvester in February this year. And in a short span of time, we have kind of grown into a full-fledged, bona fide neuroendocrine cancer program, where we have weekly neuroendocrine tumor board. We have now seen well over 240 new patients in this time frame, and presented well over 140 patients at our tumor board, weekly neuroendocrine tumor board. And and we are working very closely with our surgery, endocrine, nuclear medicine, and interventional radiology, and so on and so forth, because neuroendocrine cancers like sarcoma is truly a multidisciplinary disease and needs everybody on board. for the best possible patient care. Thank you so much for your time. I'm happy to take any questions. I'll stop sharing my screen.
Thank you, Dr. Chauhan. That was very didactic, very interesting. We appreciate it very much. As a matter of fact, I call everyone's attention to this week's New England Journal of Medicine. There's a very interesting case report of a patient with a neuroendocrine tumor. And unfortunately, they didn't hear your lecture. All they treated the patient with was octreotide. But the presentation was quite interesting of the neuroendocrine tumor with the multiple system, respiratory and cardiac system effects. So, but that's just a plug for how important your work is and this area is. Most of these, or this particular therapy that you refer to is good at containing the disease, but it doesn't. I don't see the information showing that it actually cures it. It can reduce tumor burden, but it's not cidal to the sense that you've cured them. And that's a good, and for business, perhaps maybe that's good, but not for the patient, right? Because we really do want to cure these patients. And so, what is the main obstacle or hurdle you think in terms of developing cidal therapies that will truly cure neuroendocrine tumors?
That's a fantastic question, and thank you for that. We as a field are trying to striving for a cure, and I'm optimistic, and that's the reason I got into oncology. I know a lot of people feel jaded about the oncology. Most of the cancer we currently in metastatic disease we don't have cure for, and that's including some of the heavily studied cancers like breast cancer, colon cancers. However, we are very optimistic, and there are drugs currently which have potential to cure, if not all, but at least some subset of patients. For example, with advent of immunotherapy and immune checkpoint inhibitor therapies, we all have patients in our practices who are going 5, 7, 10 years with no evidence of disease, and they had incurable cancers, and they responded beautifully to immunotherapy. In fact, just before coming for this grand round, one of my patients came from Kentucky, no evidence of disease. Statistically, he should have been dead in 6 months because he had a higher grade neuroendocrine cancers, right? So those are the kind of things we are seeing with novel, promising immunotherapies. Now, moving forward for neuroendocrine cancers, you're absolutely right, sir. We don't have curative therapies right now, but there are a lot of interesting drug development happening. For example, we are working on an investigator-initiated study where we are combining an oncolytic virus, which has affinity to target neuroendocrine tumor cells and complements beautifully with immune checkpoint inhibitors. We presented mouse model data last year at NANETS, and now we have secured funding to do first in human phase 1 study. So we're hoping in 5 to 6 months after we get FDA's blessing, we will probably target. And there in mouse models, we did induce cures. So that's one way to kind of approach it. We are also going to be opening shortly industry studies which are targeting a novel target called DLL3. It's a notch pathway ligand heavily expressed in these high-grade neuroendocrine cancers like small cell and large cell neuroendocrine cancers. And we are targeting it with what's called a BiTE antibody, bispecific T-cell engager. So basically it has 2 arms. One arm latches onto the cancer cell on the DLL3, which is expressed in the cancer, and the other arm latches onto the T cells, brings them closer to. So it's basically making your own immune system fight against the cancer. We're bringing them in the close proximity. And I was part of some of the phase 1 studies, and phase 1 studies now in public domain. Data looks very promising. Those who really respond well do really, really well. I have a personal example, as a patient, small cell lung cancer patient, again, 4th line, and 4th line, nothing really works. They basically, median survival at 4th line onwards, like a couple of months to 3 to 4 months. So the daughter of this patient, who's a pharmacist, found us through clinicaltrials.gov. And that time, this DLL3 study was just launching, and her words were, "Can you keep my dad alive for 4 months so that she can see the grandchild?" And now we are a year and a half later, and patients in complete response. So those are the kind of experiences we are seeing with novel, promising immunotherapies. Now, I wish that we can see them in more of our patients, right? So we are still very early, and we are trying to find ways how and why some patients respond so well, and how we can replicate in wider. So we are working hard on that. And then hopefully, Dr. Weiss, next year, if you do invite me for another talk sometime, I might have some better news that we are curing some of these patients.
Thank you. Dr. Oseo, there's no other speaker, right? No. Okay. I see no other questions. Well, then we get off a little bit today earlier. And I thank everybody for participating. Pay attention to the chat for the link for CME and MOC credit. And stay well. And if you need anything, don't hesitate to give me a call. Take care, everybody.
Thank you, everybody. Appreciate it.